Hello students, welcome to the 45th lecture of Physical Pharmaceutics BP 302T. I am Dr. Sushma Verma, Associate Professor in the Department of Pharmaceutics, Pharmacy Institute, NIT Greater Noida. In our previous lectures, we have discussed buffer capacity and buffer equation. In this lecture, we will be discussing the various biological and pharmaceutical applications of buffers. Right. In the initially we have discussed the application of buffers. In this lecture, we will be taking the biological applications. So, where buffers occur biologically and what is their significance while deciding the dosage formulations for pharmaceutical systems. Buffers in the biological system. Right. The human body is composed of various fluids, right. You find different types of fluids in the human body. To name a few, we have blood, right. We have lacrimal fluid, we have urine, we have saliva. So, we find different pH in the different parts of the human body, right. So far as the blood is concerned, it is one of the important fluids that we have to take care, because a number of formulations are designed so as to be directly given into the blood, right. So, if the pH of that particular formulation is not maintained, that can cause a reaction, that can cause a harmful reaction to the human body. Therefore, the maintenance of the human blood pH is very important, right. For that, we need to have an idea what is the pH of the blood. The pH of blood is maintained at about 7.4. right. So, we know that we have the pH of the blood is 7.4 and it is near to the neutral pH, right. So, 1 to 7 is the range of the acidic pH and 7 to 14 is the range of the basic pH, right. And the buffer the pH of the blood is 7.4. So, it has to be buffered so that the pH of the blood is to be maintained, and the buffers are natural in case of blood. So, it is maintained by primary buffers. These are present in the plasma. The plasma contains carbonic acid carbonate buffer and acidic or alkali sodium salt of phosphoric acid, right. So, these are the two primary buffers that are maintaining the pH of the blood, right. So, for primary we have carbonic acid and carbonate buffer and acid oblique alkali sodium salt of phosphoric acid. So, these are the two primary buffers that are used to maintain the blood pH. And second is the secondary buffers. These are present in erythrocytes, right. So, these two buffers are present in the plasma, right. And we have secondary buffers also to support the pH of the blood. These are present in the erythrocytes. Erythrocytes are red blood cells. Right, which are oxyhemoglobin and hemoglobin and acid or alkali potassium salts of phosphoric acids, right. So, for maintaining the pH of the blood, we have two buffer systems, right. One is the primary, that is acting in the plasma of the blood, right, and these are carbonic acid and carbonate and the acid oblique alkali sodium salt of phosphoric acid, right. These are acting in the plasma, and secondary buffers, these are present in the red blood cells and which are the oxyhemoglobin and the hemoglobin, and second one are the acid oblique alkali potassium salts of phosphoric acids. So, these two type of buffers are there naturally present in our body to maintain the pH of the blood to 7.4, which is near to the neutral pH, right. Second biological system is the no, it is for the blood also. Values of buffer capacity of blood ranging from 0.025 to 0.039 gram equivalents per liter per pH unit have been reported in the literature, right. So, this is the buffer capacity of the blood, right, and the physiological pH range of blood is 7 to 7.8. when the pH of the blood is decreases or rises beyond this range, life is in danger, right. So, from this we come to know that the range of the blood pH is 7 to 7.8 and the blood has also got some buffering capacity, right. If there is a change in the pH of the blood, it has got some buffering capacity, so that the pH of the blood can be adjusted, but if the pH of the blood goes beyond 7 to 7.8. So, the life of the person is in danger. So, it is very very important for us to maintain the pH of the human blood, right. Next is the pH of the lacrimal fluids or our tears, right. So, the pH of lacrimal fluid or tears is about 7.4 with a range of 7 to 8. Tears have been found to have a great degree of buffer capacity and dilution of up to 1:115 with neutral distilled water is possible before an appreciable change in pH is noticed, right. So, the pH of tears is about 7.4. So, it is again near to neutral, right. So, these aspects have to be taken into consideration while we are formulating different formulations for different route of administration, right. So, if we are going to formulate a drug for the eyes, we have to consider the pH of the tears, because if it is near to 7.4, it will be easily accepted by the eye, right. So, if it is not 7.4, that can cause an irritation into the eye, although the tears have a good buffer capacity, right. They can accommodate a range of, they can means the solution that you put into the eye, it can be diluted up to 15%, right. And before there is an appreciable change in the pH of the tears, right. So, the tears have got a great buffer capacity, great degree of buffer capacity. You can put formulations of different pH into the eye, and so it will not harm the eye, but it is advisable to give a formulation that is of the range of 7.4. Next is the pH of the urine. The average pH of urine is about 6 with a range of 4.5 to 7.8, right. So, here you find that the pH of urine is 6, but the range up to which it is maintained is 4.5 to 7.8. When the pH of urine is altered beyond this range, remedial action is taken by the kidneys in the form of retention or excretion of hydrogen ions in order to maintain the pH within the range, right. So, this is the range. So, this range is very wide, right, 4.5 to 7.8 as compared to the range of the human blood, which was 7 to 7.8, right. So, you see that the range buffer capacity of the blood is less as compared to that of the eyes and the urine, right. So, we have to be quite cautious while designing the parenteral formulation, which are directly instilled into the blood, right. So, we should be very particular about the pH of the formulations, because once it is in the blood and if the pH is not maintained, so life of that person is threatened, right. But in case of your eyes and urine, you see the range of your pH is wide, it is from 4.5 to 7.8. So, if it is beyond that range, so the kidneys automatically work, right. It can either retain the hydrogen ion or it can propel the hydrogen ions out of the body, so as to maintain the pH of the urine, because if it is not done, the body is not able to withstand it and there can be some harmful effects to the body. So, these are the three biological systems whose pH is to be maintained, and these things should be taken into consideration while formulating the formulations, right. So, pre-formulation studies should be carried out very carefully in case of dosage forms which are to be instilled into the eye or which are taken directly into the parenteral system, right, or into the blood, or which are for the kidney, right. So, you have to be very cautious for as far as the pH is concerned while designing dosage form for these very systems. Next is the buffers in pharmaceutical systems, right. So, in pharmaceutics we prepare different dosage forms, right, and different dosage forms are there, and we have to see the various pre-formulation studies before designing that. So, in case of parenteral preparations, that is injections, pH should be considered carefully as large deviations of pH may lead to serious consequences. The ideal pH of a parenteral product is 7.4, which is the pH of the blood. The most commonly, right. So, why it should be done? The pH of the blood is 7.4, right. We have just studied that the pH of blood is 7.4. So, if the formulation that we are going to inject into the blood is not of the same pH, what will happen? The body can be in danger because the pH is not maintained, then untoward reactions in the body starts, and even the death of the patient can be there, right. So, we have to be very cautious, and as we have discussed, the buffer capacity of the blood is not so good, and the range of the pH that is to be maintained for blood is very narrow. It is 7.0 to 7.8. So, the range which has to be maintained is very narrow in case of parenteral formulation. So, we have to be very cautious regarding the pH of the formulation that is to be injected into the blood. And for that, if the drug itself is of the same pH as that of the blood, then it is very good, right. We need not to put any buffer, but if that is not the case, we need to add some buffers so as to maintain the pH of the dosage form with respect to the pH of the blood. So, the most commonly used buffers in parenteral products are acetate, phosphate, citrate and glutamate. So, these are the buffers which are generally used for the formulations which are to be injected into the blood directly for maintaining the pH of that particular formulation. Next is the case of ophthalmic preparations, right. So, this is the second very important area that has to be analyzed while formulating a dosage form for the eyes, right. So, the eyes are very sensitive, these are the organs with which we see. So, the pH, you know, we have discussed in the biological systems that the pH is around about 6, right, but the positive point here for designing a dosage form is there that the buffer capacity is very good. The buffering capacity in case of ophthalmic preparations of the tears is very good. So, you can make use of that concept while designing the dosage form. So, buffers are used to maintain the pH within the physiological pH range of lacrimal fluids, that is the eye fluids. The lacrimal fluid has a pH in range 7 to 8, right. So, it has got a pH range of 7 to 8, but the thing that is good here is it has got a very good buffering capacity and can tolerate preparations having pH values between 3.5 to 10.5, right. So, eyes can tolerate formulations which have got a pH between 3.5 to 10.5 और with little discomfort. थोड़ा irritation होगा शुरू में eyes में जब eye drop आप डालते हो, but since the buffering capacity of the tears is very good, they can accommodate different pH. Outside this range, that is 3.5 to 10.5, increased lacrimation may occur with other complications. So, the buffering agents most commonly used in ophthalmic preparations include carbonate, phosphate, acetate, etc. राइट. सो आप जो है आइज़ में भी जब डोजेज फॉर्म डिज़ाइन करते हैं, कोई भी डोजेज फॉर्म आइज़ के लिए डिज़ाइन करते हैं, यू शुड कीप इन योर माइंड दैट यू हैव टू कीप द पीएच ऑफ दैट फॉर्मुलेशन कंपैरेबल टू दैट ऑफ द आइज़, राइट. ठीक है, आप जो आइज़ का पीएच है उसके हिसाब से ही आप अपनी ड्रग का पीएच मेंटेन करके रखेंगे, नहीं तो आइज़ के अंदर इरिटेशन हो जाएगा. और अगर आइज़ के अंदर इरिटेशन है तो आइज़ आर वेरी सेंसिटिव organs of the body, है ना. तो आपकी विजन ब्लर हो सकती है, any other discomfort can be there. So, you have to keep in mind that the pH of the formulation should match to the pH of the eye or the tears, though their buffering capacity is very good. पर उसके बाद भी जो है आपको एक रेंज मेंटेन करनी है एंड दैट रेंज इज 3.5 टू 10.5. उसके अगर बाहर आप गए तो इट कैन बी हार्मफुल फॉर दी आइज, एंड फॉर दैट यू कैन यूज बफर सिस्टम्स दैट आर डिस्क्राइब्ड. Next is tablet formulations. Buffer have been used in tablets and capsules to control the pH in the microenvironment surrounding drug particles. That is especially harmful in cases of drugs where the absorption is dissolution rate limited from the unbuffered formulation. Buffer have also been employed in formulations containing acidic drugs to reduce gastric irritations. They are used in solid dosage forms including antacids, sodium bicarbonate, magnesium carbonate and sodium citrate, right. So, tablet formulations में बफर हम कहाँ कहाँ यूज़ करते हैं. एक तो जब you know the tablets are taken by oral route where they get disintegrated, and after disintegration they dissolve, and the dissolved material is then absorbed throughout the GIT at a specific absorption window, right. And so you have to see that the effect in the dosage form in tablet formulations, the buffers are helpful where the absorption is the dissolution rate limited step, right. So, if the absorption is dissolution rate limited step, then from an unbuffered formulation, ठीक है. तो कोई अनबफर्ड फॉर्मुलेशन आप टैबलेट की ले रहे हैं और उसका एब्जॉर्प्शन आपको अगर कराना है, क्योंकि जब तक ड्रग जो है एब्जॉर्ब नहीं होगी तब तक जो है आपको उसका फार्माकोलॉजिकल एक्शन नहीं मिलेगा, ठीक है. तो उसकी एब्जॉर्प्शन कराने के लिए यू नीड सम buffered, or you need some specific pH, and for maintaining that specific pH you add the buffer solution. So, buffer solution वहाँ add आप करते हैं ताकि उस ड्रग का एब्जॉर्प्शन हो पाए, एंड इफ दैट पर्टिकुलर ड्रग गेट एब्जॉर्ब्ड ओनली देन इट विल बी एबल टू शो इट्स फार्माकोलॉजिकल एक्शन. Another way in which buffers have been used in tablet formulations is to reduce the gastric irritation, ठीक है. बहुत सारी एसिडिक ड्रग्स हम यूज़ करते हैं, उनको जब कंज्यूम करते हैं तो वो जो है एसिडिटी कॉज कर देती हैं, है ना. एसिडिक ड्रग्स हैं, अंदर जाएंगी, स्टमक में हमारे HCl प्रेजेंट है तो गैस्ट्रिक इरिटेशन हो जाएगी. तो उसको इरिटेशन को रिड्यूस करने के लिए हम क्या करते हैं, बहुत सारी ड्रग्स प्रिस्क्राइब करते हैं एंटासिड टाइप की, जिनके अंदर हम बफर्स यूज़ कर सकते हैं, राइट. सो दिस इज़ द प्लेस वेयर बफर्स हैव बीन यूज्ड इन pharmaceutical tablets. Next is in ointments and cream. Ointments and creams are for local application. So, you while formulating the different ointments and creams, you should see the pH of the skin which is to be maintained, right. तो आपको ये देखना है टॉपिकल प्रेपरेशंस के लिए कि स्किन का पीएच कितना है. अगर स्किन के पीएच के बराबर आपकी ऑइंटमेंट और क्रीम्स का पीएच होगा तो आप उनको इजीली यूज़ कर पाएंगे, कोई इरिटेशन नहीं होगा, right. So, topical products which are used on skin such as ointments and creams are also buffered to ensure stability of the formulation. They are either added to ensure the stability and the patient comfort. The most commonly used buffers in ointments and creams are citric acid, its salt, phosphoric acids and its salt, ठीक है. ये हम buffers use करते हैं ointments and creams के लिए. Next is the preparation of buffers, right. सो प्रेपरेशन के लिए बफ़र की हमको क्या करना है, हमें बहुत सारे बफ़र्स चाहिए फार्मास्यूटिकल एप्लीकेशंस के लिए, तो उनको बनाते कैसे हैं। सबसे पहले हम क्या करेंगे, जो है उसका देखेंगे कि वो किस पीएच के लिए हमको यूज़ करना है, राइट। द फर्स्ट स्टेप फॉर मेकिंग अ बफर इज़ टू सिलेक्ट द पीएच फॉर विच इट हैज़ टू बी यूज्ड फॉर, राइट। तो जब वो हमको पीएच समझ में आ गया किस पीएच पे उसको यूज़ करना है, तो उसी pKa वैल्यू का हम एक वीक एसिड सिलेक्ट कर लेते हैं, राइट। तो फर्स्ट स्टेप इज अ वीक एसिड शुड बी सिलेक्टेड हैविंग अ pKa approximately equal to the pH at which the buffer is to be used. This will give the maximum buffer capacity, है ना। तो बफ़र कैपेसिटी को मैक्सिमम मीन्स वो जो रेजिस्टेंस है बफ़र की, जो पीएच की, उसको वो केटर कर पाए, राइट। फॉर दैट आपको क्या करना है, तो पहले जिस पीएच को मेंटेन करना है वो पीएच सिलेक्ट कर लीजिए, और फिर उसी पीएच को मैच करते हुए एक वीक एसिड सिलेक्ट कीजिए जिसका कि pKa इज इक्वल टू द पीएच रिक्वायर्ड, राइट। And the ratio of the conjugate salt and acid required to attain the desired pH may be calculated from the buffer equation, right. So, you can use the Henderson and Hasselbalch equation to calculate the concentrations of the conjugate salt and acid required to get the pH, right. तो आप pH 4 to 10 रेंज का जब बफर बनाना चाह रहे हैं तो हैसेलबैक इक्वेशन आप अप्लाई कीजिए। पीके आपके पास है, राइट, पीएच आपके पास है, सो आपको पता है एसिड आपने ले लिया, उसकी कंसंट्रेशन कितना होना चाहिए और उसका कॉरस्पॉन्डिंग बेस जो है कॉन्जुगेट सॉल्ट, सॉरी, जो आपको लेना उसकी कंसंट्रेशन यू कैन कैलकुलेट विद द हेल्प ऑफ द हेंडरसन एंड हैसेलबैक इक्वेशन विद इन द pH range of 4:10, and individual concentration of the buffer salt and acid should then be considered to obtain the desired buffer capacity. Usually a concentration of 0.05 to 0.5 moles is sufficient, corresponding to a buffer capacity of 0.05 to 0.1, right. तो आप उनकी कंसंट्रेशन चूज कर लेंगे 0.05 से 0.5 मोल्स की, ताकि उसकी बफर कैपेसिटी 0.01 हो और 5 मोल्स की 0.1 बफर कैपेसिटी के लिए, राइट। And other factors include the easy availability of the buffer components, their compatibility with the drugs, stability of the buffer and the drug on aging, freedom from toxicity, sterility of the final solution if required, and cost of the material. So, these are the other factors that you should consider while formulating the buffers. Mainly for the calculation part, that what you have to do is you have to select the pH for which a buffer is to be prepared, right. And next step is you search for an acid, weak acid, which is having a pK value equivalent to that of the pH required. Then you calculate the concentration of the salt and the acid with the help of the Henderson-Hasselbalch equation, and then you form the buffer. And finally, you have to maintain the pH of the buffer with the help of NaOH and OHCl, right. And finally, the pH and buffer capacity of the prepared system should be determined experimentally using a reliable pH meter and pH indicator papers, since theoretical values may differ slightly from the experimentally determined ones, right. So, this was all regarding the applications, pharmaceutical applications of buffers, the biological applications of buffers, and the reference for the same are the Physical Pharmacy by Alfred Martin, 3rd edition, right. And this was regarding this. In my next lecture, we will be focusing on isotonic solutions, right. Isotonicity of solution, why it is important, and what are isotonic solutions. For that, I like to mention 2 other solutions which are used, right. So, isotonic solutions are those which are having the same tonicity as that of the physiological fluids that we are aiming at. We are aiming at formulating dosage forms for the various physiological fluids of the body. For that we have just here a look of the various physiological fluids, right. We have seen that what is the pH of different physiological fluids, and we have seen the significance of the pH of these physiological fluids. If the pH of the formulations which we are formulating for that particular fluid is same as that of that fluid, then it is okay. Otherwise, there can be some drastic issues regarding that, and the human body cannot survive in that particular pH. There can be some discomfort to the patient, and if the formulation is for the blood, the pH should be mandatorily be maintained. If it is not maintained, then the life of the person can be in danger, because the range of the pH that is to be maintained for the blood is 7 to 7.8. So, we have to focus on that while formulating different parenteral formulation. If it is not there, then the life of the patient can be at risk. So, it is of paramount importance that the formulation should be having the same pH as that of the physiological fluid. So, in my next lecture, we will be just focusing on the isotonicity of fluids and how to maintain the isotonicity, and we will be focusing on the hypertonic and hypertonic solutions, right, and I will be discussing the various methods to maintain the isotonicity of fluids, right. So, thank you so much for connecting. We will be just meeting in our next lecture for isotonic solutions. Thank you so much.